Hello students, this is Ambuj sir. In continuation of lecture 1 of chapter 2, that is sexual reproduction in flowering plants. Today we will focus on the sex organs of a typical flower, because sex organs are actually responsible for the formation of gametes, and these gametes take part in the sexual reproduction. So today's topic will be pre-fertilization, structure and theories. Students, when we talk about the male sex organ of a typical flower, then obviously it is the stamen. So this is an illustration of a typical stamen. In this stamen, dear students, you are observing this is a globular portion at the top, and this is the filament like structure. This globular portion is called anther, and this stalk like structure is called filament. And students, you are well aware that this filament is attached with either thalamus or the petal. Now you are also observing here this greenish part, which is called as connective. This connective actually connects the two lobes of a typical anther. Actually, anther has two lobes. This is a lobe, and this is a lobe, and these two lobes are connected by this tissue, which is called connective. In this way, a typical anther has three parts. A typical stamen has three parts: stalk-like filament, then anther, and the tissue which connects the two lobes of anther, it is called connective. So these three parts are there in a typical anther. Again I am repeating: filament, anther and connective. Now what is inside an anther? So to find out what is inside an anther, we cut the section of this anther. So in the transverse section of this anther, the structure which is visible is somewhat like this. This is the transverse section of an anther. In this you are observing that four corners are there. This one, then second corner, third corner and fourth corner. These four corners, they actually have a sac-like structure which is called microsporangium. Singular is microsporangium and plural is microsporangia. And inside these microsporangium, actually there is the formation of the microspores, which later on develop into the pollen grains. And inside the pollen grains, the male gametes develop. Now let us discuss the different parts of this transverse section of anther. So students, what you are observing, this, the outermost layer, which is yellow in color I have drawn here, this is the common layer, or you can see the boundary which is surrounding the anther. This is called epidermis. Now inner to this epidermis, one more layer is there, which is called endothecium. This endothecium I have drawn in pink color. So this endothecium and this epidermis, they are actually form the covering of the anther. The outermost covering is the epidermis, and inner to that there is a covering of endothecium. Now the cells of epidermis are barrel shaped and flat, whereas the cells of this endothecium, these are columnar with with fibrous thickenings. Now inner to this, a brown layer is there, which is called middle layer. Middle layer, actually it is made up of 2 to 3 layers, and just the innermost layer of this microsporangium, you can say, that is the tapetum. Tapetum, this layer provides nourishment to the developing microspores. So again you listen, I have told the function of this tapetum, no doubt, but one more thing is there, very important regarding this tapetum, that is, the cells of the tapetum, they have more than one nucleus, and these nucleus they form because of the endomitosis. Now what is the purpose of more than one nuclei? Definitely it is so that it can provide nutrition in the most efficient way. Now the outer 2 layers, that is the epidermis and the endothecium, and one more, yes, this is the middle layer. Out of 3 layers, these layers are collectively responsible for protection, and as I told you, the innermost layer is responsible for the nutrition because it provides nutrition to the developing microspores. Now this is a detailed structure of a microsporangium. So in this, actually this is a cellular diagram, this is an outline, but this is a cellular diagram. Here you are observing the outermost layer of the microsporangium, you can see. This outermost layer, as I told you, obviously it is the epidermis, made up of the barrel-shaped cells which are flanked. Inner to this endothecium, here the cells are visible. These cells are columnar with fibrous thickenings. Actually these fibrous thickenings, they help in the dehiscence of the anther. You are observing there are few cells here, they don't have the fibrous thickenings. Actually this region forms the stomium. Here I have mentioned here stomium. Stomium is responsible, stomium is that part of the anther from where the dehiscence takes place and the pollen grains they shed. Now just inner to this endothecium, 2 to 3 layers are there which collectively form the middle layer. Now here I have drawn 2 layers, 1 and 2. These middle layers, this epidermis, endothecium and middle layers. Together these 3 layers are responsible for the protection. They protect the pollen grains which develop inside the microsporangium. Now the innermost layer, which I have drawn in orange color, this is the tapetum, and tapetum is responsible for providing nutrition to the developing pollen grains which later on develop into the developing microspores. These microspores later on develop into the pollen grains. Now at the center here is a group of cells. These group of cells, they together form the sporogenous tissue. Sporogenous tissue are actually responsible for the formation of microspores, and these microspores later on develop into the pollen grains, and inside the pollen grains the main gametes develop. Now, students, just try to understand, when anther is in its initial stage, it is a mass of parenchymatous cells somewhat round like this. Not round, it is oval. This is the epidermis. Epidermis. And in the center, there is group of parenchymatous cells. These are the parenchymatous cells. These are the nuclei, parenchymatous cells. Right? Later on, this oval structure, this is the initial stage of the anther, means the anther is coming into the existence. So that time it looks oval, and it is just a mass of parenchymatous cells surrounded by a epidermis. Now, after some time, four corners develop like this. One is this one corner, next, and this is the last corner. And each corner, few cells, they differentiate. This will be the epidermis. Right? So, this is as usual the mass of the parenchymatous cells. Now, students, some hypodermal cells, they differentiate because of their bigger size, dense cytoplasm, and bigger nucleus. Hypodermal cells means below the hypodermis. Like some cells here differentiate. Supposing this is a cell, it is differentiated. One more cell differentiated. Here is a cell differentiated because of its bigger size and dense cytoplasm. Here again, one cell. So at each corners, few cells become differentiated because of their bigger size, dense cytoplasm, and prominent nucleus. Now these cells are called as archisporal initials. What? Archisporal initials. ARCHESPORIA. Initials. Archisporal initials. These archisporal initials later on divide by pericranial division. Actually, pericranial division is a type of mitosis, but it is the matter of the plane. Because of that, the name is given on which plane the division is taking place. So, because of the pericranial division, each archisporal initial, it gives rise one cell towards the epidermis and one cell towards the center. The cell which is towards the epidermis, that is called primary parietal cell. What do we call? Primary parietal cell. Primary parietal cell. And the cell which is towards the center, that is called primary sporogenous cell. Right? Now this primary parietal cell further undergoes pericline division and it gives rise the endothecium and middle layer, and also this tapetum. Endothecium, middle layer and tapetum, they come into existence because of the division in the primary parietal cell. So primary parietal cells, they are responsible for the formation of the wall layers of the microsporangium. Now this primary sporogenous cell, it also undergoes further mitotic divisions and it gives rise a mass of cell which is called as sporogenous tissue, that I have drawn here, sporogenous tissue. So in this way there is the anther becomes mature, or you can say the microsporangium, they come into existence. What important is that, which we must understand, that in a typical anther there are two lobes. One is this one. This is a lobe. This is, I have drawn one lobe, this one, and this is the another lobe. So this is this side, one lobe. One lobe has two microsporangia. This another lobe. Again it has two microsporangia. So in a typical anther, four microsporangia are there. Anybody will tell me the example where there is only two microsporangia is there in a typical anther? Yes, there is a family which is called Malvaceae. In Malvaceae family, the anther has only one lobe, and since their one lobe is found, so only two microsporangia are there. Right, students? So now this type of anther is called tetrasporangiate. A technical word is there, you must learn it. Tetrasporangiate. Tetrasporangiate. What is the meaning of tetrasporangiate? Means an anther which has four microsporangia. A typical anther is tetrasporangiate. Now one more technical word is there, you must learn it. Eusporangiate. What is the meaning of eusporangiate? Anther is eusporangiate. What does it mean? Meaning the microsporangia, they develop from group of cells. I told you that some hypodermal cells, they differentiate because of their bigger size, dense cytoplasm and prominent nucleus. So some, not one, more than one, means a group of cells are responsible to form the microsporangia, and this condition is technically called, that type of anther is called eusporangiate. So I think you must clear right now what is the meaning of tetrasporangiate anther and what is the meaning of eusporangiate anther, right? Now we will proceed with the next topic, that is microsporogenesis. Students, in the center of microsporangium, what we have observed? Yes, a group of cells is there, which forms the sporogenous tissue. Now, in a mature anther, each cell of the sporogenous tissue that becomes a microspore mother cell. That is a microspore mother cell. Supposing it's a microspore mother cell. Just a moment. It is the microspore mother cell. Microspore mother cell. It is the microspore mother cell. Now, in this microspore mother cell, a very peculiar type of division takes place. Students, you are well aware that two types of the cell divisions are there. Very famous. You have studied in class 11. One is called mitosis, and the other one is called what? Meiosis. So, during the sexual reproduction, which cell division takes place? It is meiosis. So, this microspore mother cell, it undergoes meiosis, and as a result, there is the formation of what? Microspores. What? Microspores. So, these microspores, when a cell undergoes meiosis, so how many daughter cells are formed? Yes, 4 daughter cells are formed. So, these are 1, 2, 3, and one is behind this, which is not visible, right? So 4 daughter cells are formed here. This is called, these are the microspores. Since these microspores are attached with each other, that is why a technical word is given here, which is called tetrad. So here you can write microspore tetrad. Now this microspore tetrad, when the anther becomes mature, then these microspores, they separate, means separation takes place. Now 1, 2, 3, 4. What are these? These are called as pollen grains. So in this way, the pollen grains are formed. Right? So the microspore mother cell undergoes meiosis, then microspore tetrad formation takes place, then separation takes place, and the microspores, they develop into the pollen grains. Now, pollen grains are the first cell of the male gametophyte. Male gametophyte. I'm writing here, male gametophyte. Male gametophyte is it's again a technical word. So if anybody asks from you what is the first cell of the male gametophyte, you will say it's pollen grain. Now a question comes here, what is the meaning of gametophyte? So gametophyte, this word or term has two root words. Two terms are there. One is called gameto and the other one is called phyte. Phyte. Gameto is a derived word. The meaning of this which tells us about the gamete. means the root word is here gamete. Gamete is a derived word. For this the root word is gamete. And phyte is again a derived word, and not derived, it is just a term, and the phyte tells us about the plant. Meaning of phyte is here the plant. So now we can understand that part of the plant which is actually responsible for the formation of gamete, that plant part which is actually responsible for the formation of gamete is called what? Tell me. It is called pollen grain. So pollen grain is that part of the plant in which there is the formation of the male gamete. That is why the pollen grain is called the first cell of the male gametophyte, right? Sorry, pollen grain. No, it is some confusion here. Just I'm giving you the correction. The microspores are the first cell of the gametophyte. Sorry. So microspores are the first cell of the gametophyte, and these microspores, they become the pollen grain, and pollen grain is actually the gametophyte. Inside that there is the gametes are formed. Okay. Now let us concentrate the structure of a pollen grain. So students, this is the pollen grain. Right? So in a pollen grain, this is the outermost layer. No, it is not the outermost layer. Above it, one more layer is there. This is one more layer. Right? Just see this outermost layer. This is called sporopollen. Yes, sporopollen is a very important term. More often the questions asked from this sporopollen. Sporopollen is a very hard substance, and it is there is no impact of temperature. High range of temperature cannot affect this sporopollen. There is not any impact of the acid or alkali on this sporopollen, and so far there is not any enzyme known which can degrade this sporopollen. So it's very hard substance, and that is why the pollen grains are found in the fossil form because of this sporopollen. Now, sporopollen is not a continuous layer. It's not a continuous layer. I mean, this is a depression you are observing. Again a depression is here. Again a depression is here. So here the sporopollen is absent. At the depressions, the sporopollen is absent, and these depressions are actually called germ pore. What do we call? Germ pore. Right? Now, this sorry, just listen. Actually, this is the outermost layer. It is called exine. Outermost layer of the pollen grain. It is called exine, and this is the innermost layer of the pollen grain. It is called intine. Right. Now, exine is made up of sporopollens. This you will learn. Exine is made up of sporopollen, and intine is made up of pectocellulose. Pectocellulose means pectin plus cellulose, right? Now, this is a depression which is called germ pore, and at the depressions, the exine is, absent. Only intine is there, right? Now there is a third layer. What do we call this layer? Yes, anybody tell me. This third layer, what do we call this third layer? It is called, yes, plasma membrane or cell membrane. Plasma membrane or cell membrane. Now, this area which is occupied by the plasma membrane, in this liquid is formed, and that is called cytoplasm. Now, this cytoplasm is differentiated into two cells. One is the bigger cell, this one is the bigger, and this one is the smaller. The bigger cell, it is called vegetative cell. And the smaller cell is called generative cell. Since these are the cells, so we will draw here the nucleus. The nucleus. This is also a nucleus. So the bigger cell is the vegetative cell, and the smaller cell is the generative cell. Vegetative cell, this forms the pollen tube in future at the time of germination, and this generative cell is responsible to form the male gametes. So this is the structure of a pollen grain. It's a pollen grain. What is the ploidy of pollen grain? It's a N, because pollen grains, they come into existence because of the meiosis in the microspore mother cell. So what will be the ploidy of the microspore mother cell? It will be the 2N. What will be the ploidy of the microspores? It will be N. And later on, whatever structure is formed, that will have the N ploidy. So pollen grains, they will also have the N ploidy. Now, because this cell is haploid and this cell is haploid, so it's also the ploidy is N. Means it's a haploid structure, pollen grain. Now this, in 60% situations, or in 60% conditions, the pollen grain is shed in this two cell stage, and it reaches the stigma of the flower, right? But it has been also observed that in 40% conditions, the pollen grains, they are shed in 3 cell states. What does it mean? This generative cell, it divides mitotically to give rise to 2 male gametes, right? So 2 male gametes plus 1 vegetative cell, now 3 cells. So in this way, in 40% conditions, the pollen grains are shed in 3 cell states, but in 60% conditions, the pollen grains are shed in 2 cell states. That time, when this pollen grain reaches the stigma, then germination starts, and when the germination starts, then this vegetative cell come out from a germ pore in the form of pollen tube, and in this pollen tube, this generative cell move, and on the way, I mean in the pollen tube itself, it divides to give rise to male gametes, right? In this way, the pollen grains, they shed in 2 cell states, I mean in maximum 60% conditions, and 3 cell states in 40% conditions. So students, this is all about today's lecture. Thank you very much.